All right, the final chapter we're going to cover is chapter 19 on viruses. I omit 20 and 21. Normally I'll go over those two chapters together, but we cover a lot of that information in lab. So jumping into chapter 19, viruses. As you see there, the reason for the zombies. First semester I taught this course here at ACC. I had a lot of students that were very, involved with The Walking Dead. So every Monday, all they talked about was The Walking Dead, and it was driving me nuts. But then when I got to viruses, viruses are described as not being alive, but they're also not dead. They're in an in-between state or in-between chemical state. And I kept thinking, what could I use as an everyday representation? And it occurred to me, zombies. You know, they're not alive, they're not dead. If they get close to you, they can transform you, causing you harm. So that pretty much sums them up. So the figures up here that I'm showing you are the Ebola virus, influenza virus, and HIV. All three of these are animal viruses that we'll talk about, especially towards the end of the chapter, but that's the purpose of using zombies, just to give you an understanding of how it relates to something that you're used to seeing, in this case in the movies. This next slide here is focusing on the influenza virus. Okay, these are micrographs or EMs of the influenza virus. You can see here with the influenza virus, I've also put to the side here a little pig or swine and chickens, or what we call avians or birds. Because when you hear about the flu, you often hear about the avian or bird flu, and you often hear about swine flu or pig flu. Okay, roughly back around 08, 09, we had an outbreak of the swine flu. I had a couple of students that contracted the virus, and they were kind enough to pass it on to me. Symptoms were different. I never ran a fever. I went into work the whole time, but the congestion was unbelievable to the point I ruptured an eardrum. They refer to the strain of the influenza virus that they call the swine flu as H1N1, a type A influenza virus, which we'll talk about a little bit more. Then you have the avian or bird flu. Commonly you hear about this up in China, but actually this strain of the influenza virus, okay, has made its way to the US, and I know up in the Midwest around Ohio or so, they had to, if you want to call it, euthanize, okay, these chicken farms, they had to euthanize all the chickens because there was an outbreak. So this is what they talk about. So as we go through this, we'll talk about the influenza virus and how it's an animal virus, but depending on the type, okay, that contributes to whether or not they can infect any animal or just humans. Down here, we talked about this. This is right here, the bacterium E. coli. Attached to it are many viral particles. So sometimes a virus is referred to as a viral particle. So you'll hear me say viral particle sometimes. But you can see here, it's not a one-to-one relationship, okay? When an organism is exposed to a virus, it's actually many viral particles. Okay, and many can attach to the outer barrier of a cell. Here they're showing the virus bacteriophage. You can see many of these bacteriophages have attached to the outer barrier of this bacterium E. coli. And what researchers have discovered is that often when you see this scenario here, okay, where it's not a one-to-one ratio, it's many bacteriophages attached to one cell. What often happens is that more than one viral particle will successfully insert its genetic information, thus transforming, okay, the cell into a host cell. But since the host cell, okay, has multiple copies of genetic information inserted into it, it could result in the host cell reproducing new viral particles that are recombinant. In other words, it's a new strain. So you hear about the coronavirus, but what you don't hear much about the coronavirus is that researchers have been tracking the various strains of this COVID-19. Such as up in Washington State. By the time the virus had made its way to Washington State, where we actually had our initial serious outbreak, not in New York, but in Washington State, they traced it back to two individuals that had traveled overseas. One of them had brought back what they call the Wuhan strain of COVID-19. Another individual brought back what they call the Italian or Italy strain of COVID-19. So the virus has been mutating. So what they're not telling you is that they worry about these mutations as far as causing the virus to become more infectious or not. Now, but they can actually use the genetic information in these viruses to track, okay, their origins or where they're derived from. Now here, what I've shown is, if you recall, in order to be considered a living organism, you must possess or exhibit 7 traits, 7 common properties. Viruses, there's order to them. Okay, there's a question mark by regulation. They apparently are responding to stimuli because they definitely evolve. However, there's no growth and development. Okay, there's no proof that they're consuming nutrients and using some of it to produce energy. And viruses, this is the key one I focus on. They don't have the ability to reproduce on their own. Okay, in order to be considered a living organism, you have to have the ability to reproduce on your own. Thus, this is the next slide. The next slide here is showing you how a virus requires a host cell in order to reproduce. Okay, when a cell becomes a host cell, what happens is the virus inserts its nucleic acid, its genetic information stored in a nucleic acid. In this case, it's DNA. Okay, when a virus successfully infects a cell, that means it has inserted its nucleic acid, such as DNA, which contains the viral genetic information on how to make more viral particles. The structure of the virus, okay, I compare to if you're with the Apollo space missions, how you'd have a large rocket and each part consisted of boosters that were supposed to take the astronauts to a certain elevation, eventually reaching the moon. And as they reached that elevation, piece by piece of the overall rocket would fall off. It's similar to viral particles. The structure of the virus is designed to help the virus infect the cell, inserting its nucleic acid, which contains genetic information, thus transforming that cell into a host cell, or another way to say it, a viral reproducing factory. Now that host cell, okay, which contains enzymes involved in gene expression, as well as enzymes involved in DNA replication. Well, the host cell's own enzymes involved in DNA replication and gene expression are now being used to help reproduce more of the virus. Once the new virus is assembled, it will exit the cell. Sometimes the virus will exit the host cell without killing the cell. Sometimes the virus will cause the host cell to rupture, releasing many viral particles. So here it's showing a 1-to-1 ratio as far as one virus infecting one host cell. But keep in mind, that's not actually how it works. It's many viral particles, okay, trying to infect a single host cell. Another thing I forgot to point out real quick. Notice the difference in size. You may have been hearing about filters, wear a mask, wear a mask. A lot of people, it used to be a lot, but now some people think wearing a mask protects them from becoming infected with COVID-19. No, you're protecting your neighbor. Any droplets from sneezing or coughing, okay, that would contain the viral particles, the mask helps trap them in that moisture or that droplet. Okay, so you're protecting the person next to you. So if you're wearing a mask and you're surrounded by people who aren't, you're actually the one that's not protected. Okay, so I'll show you a figure that I edited it to explain this a little more in a bit. And I'm going to show it to you now, actually. I edited this slide. I added this in. This is from chapter 6. Okay, if you look here, it shows you roughly this is where the virus is in terms of size. Okay, if you notice, viruses are very small. They're in the nanometer range. Okay, as we go up, you start getting into the area of bacteria, then you start getting right about here, and then you get into the area of eukaryotic cells, where we're now in the micrometer range. Therefore, this is where they talk about an N95 mask. Okay, the pore sizes in that mask are small enough to prevent viral particles, okay, from entering if you have the mask put on correctly and secure. So in that case, with an N95 mask, you are protected. You're not only protecting the people around you, but you're protected yourself. Okay, so they're very, very tiny particles. Leads to this next part. Much of what is known in cell and molecular biology, okay, a lot of that can be attributed to discoveries having to do with viruses. Okay, by roughly the late 1800s, scientists knew there was something out there smaller than a bacterium, okay, that was functioning as an infectious agent, causing disease in living organisms. By the early 1900s, okay, tobacco farmers, okay, were very alarmed because they were losing their crops. Instead of their crops looking healthy like here, okay, their plants that were growing had leaves that had this mosaic of patterns, this green and yellow pattern. Well, and then these tobacco plants would die. They wouldn't fully develop. They were ugly, they would die, so obviously they were losing a lot of money. So by the 1920s, they were pumping quite a bit of money, okay, into research to figure out what is causing this problem. Okay, so what happened by the 1930s is scientists had taken infected plants, tobacco plants, extracted the sap from those plants, okay, and then added that sap to a filter. Now, you can't see the filter real clearly on here, okay, but the filter is roughly right here where I put the line. It's a micron filter. Okay, when I worked in the lab, we worked with human cells. We used micron filters to filter out anything, okay, that was the size of bacterium or larger. Some of these micron filters we refer to as 0.2 or 2 micron filters, okay. The smallest known bacteria are approximately 0.2 microns. So often we would use a 0.2 micron filter. So basically what happened is they would extract the sap from these plants that appeared infected. They would put it in a container that had a micron filter that would filter out anything the size of a bacterium or larger. What they're not showing you here is they would connect this little outlet here on the Erlenmeyer flask to a vacuum, thus sucking this tree sap through the filter down here into the Erlenmeyer flask. Anything that was the size of the bacterium or larger should have been trapped in the filter. Therefore, this solution here should have been sterile. What scientists did next is they took this filtered and so-called sterile sap and they brushed it on the surface of a healthy tobacco plant or the leaves of a healthy tobacco plant. And lo and behold, what happened? That healthy plant became infected. So that let scientists know, okay, what we're dealing with is an infectious agent that's smaller than a bacterium. We don't have the technology to figure out what it is, though. So roughly by about 1935, there was a scientist by the name of Wendell Stanley. He became very popular for a new technique called crystallography. So he was provided with some of this sap from an infected tobacco plant. And he performed a technique where he was able to crystallize the infectious agent. In other words, I've got a results here that give me an idea of what I'm looking at. And he told other scientists, apparently our infectious agent, okay, is a protein. Okay, apparently we're dealing with something made up of many proteins complexed together into a large protein structure. Okay, so here you're seeing an EM, and it's similar to what he would have seen. Well, as technology advanced, researchers came to find out, well, yeah, it looks like protein in the crystal structure, but in actuality, these proteins have formed a coat called a capsid. Just know it as a capsid. You can ignore this capsomere stuff. Wherever you see capsomere, just say capsid. And what they noticed is that these proteins formed a coat called a capsid that surrounded the nucleic acid, RNA. This became known as the tobacco mosaic virus, more popularly known as simply TMV. Okay. So it was discovered it was a virus that was the infectious agent. And they knew about viruses because by then they had also been working with bacteriophages. It was just hard to work with these because of limitations in technology. But they came to find out and discover that this was a virus. So this leads me to my next part here. When it comes to viruses, okay, excuse me, viruses at a minimum, when we talk about their chemical structure, viruses at a minimum are made up of nucleic acids and proteins. If you look down here, they're giving you 4 viruses. In addition to TMV, tobacco mosaic virus, they're giving you adenoviruses, which are associated with the common cold, like rhinoviruses, influenza viruses, and bacteriophages such as T4. There are many bacteriophages. Up here, they're giving you simple diagrams. Here, they're giving you the EMs. But if you look at these viruses, for all 4 viruses, they're trying to point out to you we have a capsid or glycoproteins, okay, or a tail sheath, tail fiber, or a head. In other words, they're referring to proteins. Okay. In addition to that, if you look at all 4, they're showing you'll either have RNA or DNA present. So nucleic acids are present. For all 4 of these, at a minimum, you see they're chemically, structurally speaking, they're made up of nucleic acids and proteins. However, if you look at the influenza virus, you have one more additional compound contributing to its formation. It's called a membranous envelope. Okay, you don't see this for the other 3 viruses. Okay, now to go down the line, and I'll explain this last part in a bit. To go down the line here, nucleic acids. Viruses, okay, are initially classified as either DNA viruses or RNA viruses. And I'll show you this for animal virus later in the lecture, okay? The catch is this. If it's a DNA virus, okay, it can either possess single-stranded DNA or double-stranded DNA. And if it's an RNA virus, the RNA can be single-stranded RNA or double-stranded RNA. Now, the abbreviations I use for single-stranded is SS. So if I'm talking single-stranded RNA, I would put ssRNA. If I'm talking double-stranded, I would put DS. Okay, so single or double-stranded RNA would be SS or dsRNA. Single or double-stranded DNA would be SS or dsDNA. You can find any combination. Okay, however, the function is always the same. Okay. The purpose of the nucleic acids, that's how the virus stores its genetic information. So if it's a DNA virus, such as they're showing here for the adenoviruses and the bacteriophage, then that is the compound that the virus has its genetic information stored in. Okay, if it's an RNA virus, such here or here, then that's the compound that the virus has its genetic information stored in. Okay, it just depends. But remember, and I've said this before, for living organisms, all living organisms have their genetic information stored in DNA, and the DNA is double-stranded. This is not true for viruses, okay? So that's one thing about the nucleic acids for viruses. It can be single or double-stranded RNA, single or double-stranded DNA, but in each case, the function is always the same. That's how the virus has its genetic information stored. In regards to proteins, at a minimum, the proteins will form a capsid, a protein coat. Where it says capsamere, automatically tell yourself capsid. Okay, you can see capsid over here. For the bacteriophage, okay, they call it the head, but technically speaking, it's a capsid. It coats the nucleic acid. So when it comes to the proteins, okay, the proteins will contribute to the chemical structure of a virus by forming, at a minimum, a capsid, which is a protein coat that surrounds or encases the nucleic acid. Okay, so if you come over here to the influenza virus, it consists of 8 molecules of single-stranded RNA, and each molecule of RNA is encased in a protein capsid. Okay, now if you look here for a couple of these, when it comes to the proteins, you can see for a couple of these, they're showing you what we call glycoproteins. Okay. What has been found is in some viruses, you also have proteins contributing to forming these little needle-like structures that extend out from the virus. If you've ever seen a pin cushion, a little bunch of needles stuck into it, that's what this looks like. So each of these is sticking out, whether it's short, these little pin-looking things, each of these is a protein. Okay, it's been found that for some viruses, our immune system targets glycoproteins, and I'll go over the influenza virus in a second. If you ever hear about the influenza virus, such as swine flu, H1N1, there's a form of avian flu called H5N1 or H3N2. They're referring to the glycoproteins on that influenza virus. So, strictly speaking, though, the proteins not only can form a capsid, they can form these glycoproteins. Now, technically, these proteins have sugar, but I just want to focus on the protein. And if you over here, if it's a bacteriophage, every once in a while, you have those viruses where we don't use the terms capsid, we don't use the terms glycoproteins. Instead, we say head, tail, teeth, and tail fiber. So there's some exception when it comes to the viral world. So when you look at this, these 3 viruses, TMV, adenoviruses, and bacteriophages, they're all made up of nucleic acids with proteins. The exception comes in with the influenza virus. As I said before, the influenza virus it also has contributing to its chemical structure lipids. These lipids form a structure called the membranous envelope. In other words, they form a membrane. And what scientists discovered, okay, is that not all viruses have this. For those viruses that do, such as the influenza virus, Ebola, and HIV, they all have a membranous envelope. Okay? The structure of that membranous envelope is described as a phospholipid bilayer. Well, what ended up happening is scientists discovered that this membranous envelope comes from or is derived from the host cell the new viral particle is leaving. So this membranous envelope actually comes from the host cell to reproduce the virus. So it's like a wolf in sheep's clothing. Sometimes a membranous envelope will be derived or come from the Golgi apparatus of the host cell. But in the case of the influenza virus, the membranous envelope, the reason why it looks mainly like a phospholipid bilayer is because it's coming from the host cell's membrane as the rest of the compounds are exiting the cell. So this really put, how do you say, really threw the scientific community on its ear. It's like, oh my God, this virus is actually carrying a piece of the host cell. So this is the case for your animal viruses, such as the three I mentioned. Now, these are the general compounds, some of which I've mentioned, okay, in the influenza virus. But if we wanted to focus strictly on the influenza virus, we would start off with nucleic acids. Okay. For the influenza virus, so the way I've given you your notes is I give you some general information on the different types of organic compounds that contribute to the formation of all viruses. At a minimum, nucleic acids and proteins contribute to the formation of all types of viruses. I've never seen anything different. However, what I go transition into next is let's apply this general information specifically to the influenza virus. The influenza virus at a minimum is made up of nucleic acids. Well, specifically, it possesses 8 molecules of single-stranded RNA, which they depict here in red. Each single-stranded molecule of RNA is enclosed in a capsid. So if you go to this next figure, we kind of have to use both figures to look at it. If you go to this next figure, they don't say RNA and they don't say capsid. They say RNP. RNP stands for ribonucleoprotein. Whenever you see a term or see these abbreviations RNP or written out ribonucleoprotein, automatically tell yourself they're talking about a chemical structure of RNA and protein. So what they're pointing at here, right in here, there's better detail here. This is your ribonucleoprotein. It's made up of single-stranded RNA encased in a protein capsid, and there's 8 molecules. So the influenza virus has its genetic information stored in RNA. Next, we go to the proteins. Okay. For the influenza virus, again, we already said the proteins form a capsid, and the proteins form glycoproteins. Okay, but they don't form any glycoproteins. The proteins that are specifically made up primarily, the structures that are primarily made up of protein are these 2 that I circled. Now they're glycoproteins because there's some sugar, but I cheat a little, I just put them under proteins. Okay. For the influenza virus, not only do proteins contribute to the formation of a capsid or a protein coat surrounding a nucleic acid, but also proteins contribute to the formation of glycoproteins. 2 types of glycoproteins, hemagglutinin and neuraminidase. Hemagglutinin, it was discovered, okay, that it functions in attaching the viral particle, the influenza virus, to the membrane of what will eventually become a host cell. Okay, so this acts as an anchor. It helps attach the virus to the membrane of what will become the host cell. They often refer to hemagglutinin as a glycoprotein spike. That's why they're showing it to you this way. Now, the abbreviation for hemagglutinin is H. Okay, and it has been discovered that there are anywhere between 16 to 17 different forms of this type of glycoprotein called hemagglutinin. Down here, neuraminidase. Proteins also contribute to the formation of a different type of glycoprotein. It is not referred to as a glycoprotein spike. Okay, this glycoprotein is called neuraminidase. It was discovered the function of this glycoprotein is to help the newly produced viral particle exit the host cell. So notice they show it like little suction cups. That part I don't know why, but it's definitely not shown as spike. The abbreviation for neuraminidase is N. Okay, so if you pay close attention, especially during flu season and when they're coming out with the vaccine or when there's an outbreak of the flu, okay, often you will hear them say, like I said before, H1N1, swine flu, H5N1, avian flu. I think there was also an H3N2, which was actually described as a bird flu. But the H and the N that they're referring to, or the H and N that they're using the abbreviation, the H is for the hemagglutinin. The N is for the neuraminidase. Now, I forgot to mention for neuraminidase, there's up to 8 to 9 different forms of this glycoprotein. Okay. Now, because of these differences, okay, in part, because of these differences to the glycoproteins, influenza virus is actually broken up into 3 types, A, B, and Z. Okay. Type A influenza viruses were made up of different combinations of H and N, all right, can infect any animal. Those strains of the influenza virus that are classified under type B or C only infect humans. Now, the predominant types that circulate the world causing the flu are type A, but if you pay attention to this past flu season, especially in spring, okay, the committee that decides, okay, what the combination or what the formula will be for the flu vaccine that's given out, they decided to change it up. They decided to add components from a strain of influenza virus that falls under type B. So by the spring, the flu vaccine consisted of a mix of parts, okay, of the influenza virus that fell under type A, as well as parts of the influenza virus fell under type B, and they did a pretty good job of predicting it. But then again, that got covered up because of the COVID outbreak. But the way it works also, everybody, is normally when you hear something like H1N1 or as I said, H2N3, often they're referring to type A. But if you hear them talking about a flu outbreak and they start talking about the Kansas strain or the Brisbane strain or something like that, automatically tell yourself they're talking about type B influenza viruses. Okay. Now, the other part, though, that you see, and this one really sent the scientific community on their ear, is that unlike the other viruses I was talking about here, these other 3 types, TMV, adenovirus, and bacteriophages, the influenza virus was found that it also contained proteins that functioned as enzymes, specifically an enzyme that came to be known as Rna Transcriptase. Remember, if we're talking about a protein that functions as an enzyme, we give it a name with the last 3 letters ASE. This is prepackaged into the virus, so I'm pointing to these little molecules here, and you will find this enzyme whenever a virus is carrying an enzyme prepackaged, okay, that doesn't come from the host cell. You'll find it often right there adjacent to where the nucleic acid is encased in the capsid. This enzyme here, Rna Transcriptase, down below is its function. It performs viral RNA replication. Listen carefully, please. All living organisms have their genetic information stored in the form of DNA. If you go back to chapter 16, DNA replication, an enzyme called DNA polymerase requires a DNA template to synthesize DNA. You jump ahead to chapter 17, gene expression, the first step, transcription. An enzyme called RNA polymerase also requires a DNA template, but it synthesizes RNA. Listen carefully. Any enzyme that has as part of its name transcriptase, this is an enzyme that requires an RNA template to synthesize a nucleic acid. If the enzyme not only has as part of its name transcriptase, but the first part of its name is RNA transcriptase, then that means this is an enzyme that requires an RNA template synthesize more RNA. So in the front of it, it's telling what it's going to make. Going back again, DNA polymerase. Polymerase requires a DNA template. DNA polymerase, the DNA tells you what it's going to make, more DNA. RNA polymerase. Polymerase tells you it requires a DNA template. But the RNA tells you that RNA polymerase is going to make RNA using a DNA template. For transcriptase, anytime the enzyme has transcriptase, it requires an RNA template. Well, this is something researchers haven't been able to figure out. If a virus is not alive or dead, then how does a virus such as the influenza virus know to provide the host cell with an enzyme that is critical to reproducing the virus? The cells of living organisms do not perform RNA replication, and the influenza virus is only more than happy to say, here, I'm going to give you an enzyme called Rna Transcriptase, and you're going to perform viral RNA replication. You're going to make copies of my 8 molecules. So these are things that baffle the scientific community. You're not alive, you're not dead, but oh my God, there's so much variation in you. The last part for the influenza virus is the membranous envelope. Again, in this case for the influenza virus, this is a membranous envelope they're showing you here. Okay, it's derived from the host cell's membrane as it's exiting the cell. And I'll show you a figure a little while later when we go to the different classes of viruses. So this is the last component. Well, the membranous envelope is a phospholipid bilayer. Hemagglutinin helps attach the virus to the surface of the cell membrane of what eventually will become the host cell. It just has that hemagglutinin is helping in pulling up the membranous envelope close enough to the cell membrane, the host cell, so that the phospholipid bilayers can fuse together, thus releasing these ribonucleoproteins or molecules of RNA encased in the capsids into the intercellular environment or ICF of the host cell. So again, like I said, the way they're designed, okay, the way they're designed is in a manner that, sorry, I don't remember which way I'm going, that it works much like, if you want to call it a spacecraft. So this here is just to remind you, okay, if it's a DNA virus, once the virus inserts its genetic information, its nucleic acid, transforming it into a host cell, that DNA, okay, is going to be replicated by the host cell's own enzymes, okay? And whether it's a DNA or an RNA virus, okay, at a minimum, the ribosomes, the RNAs, okay, and anything else that's required for translation, okay, that's present in the host cell will now be used to help with the production of protein, but viral protein. So the virus is, all it's doing is saying, here, I may spot you a protein that functions as an enzyme to get reproduction started, but other than that, it's just taking advantage of everything else that the host cell already produces. So the way we talk about this, we make it seem like the cells of a living organism or a living organism that is unicellular, like they're helpless, and they're not helpless. Case in point, talked about bacteriophages. Simply put, they're viruses that infect bacteria. Again, here you have an E. coli bacterium. It's got a bacillus or rod shape to it. Each of these is an individual bacteriophage that I said before reminds me of a lunar landing craft with the tail fibers, tail teeth, and then the head up there. But what scientists discovered, okay, is not only at the multicellular eukaryotic level, but even at the unicellular prokaryotic level, okay, such as with bacteria, they're not helpless. They do have a defense system. I think everybody's heard of CRISPR. Well, CRISPR actually, okay, in vivo, in a bacterium, CRISPR is actually part of the bacterium's immune system. Okay, it's a system that takes a fingerprint of the viral nucleic acid, so that if the same strain of virus infects the bacterium again, the bacteria can fight back even quicker, much the same as with our own immune system. Okay, but prior to CRISPR being discovered, okay, at least as far back as the 70s, probably the 60s, it was maybe even earlier, it was found bacteria produce enzymes, protein enzymes called restriction enzymes, which they're showing you here. Restriction enzymes, which are depicted here with these little green dots. Restriction enzymes are proteins, okay, that digest or cut up nucleic acids, especially DNA. They primarily focus on DNA. So what they're showing you here is, let's say the bacteriophage is a DNA virus, because bacteriophages have been found to be DNA or RNA viruses. But let's say the bacteriophage is a DNA virus. Once a tail is attached to the molecules on the outer surface of the bacterium, the tail sheath will inject the double-stranded DNA in the head into the bacterium, transforming it into a host cell. But the bacteria produces proteins called restriction enzymes, which will scan the double-stranded DNA, looking for a specific combination of bases along the DNA. That specific combination of bases along the DNA is called a restriction site. So the term restriction site is referring to the DNA. The term restriction enzyme is referring to the protein that will recognize and bind to that stretch of DNA called a restriction site. And the restriction enzyme, when it binds to a stretch of DNA called the restriction site, it will cleave or cut or simply digest the DNA, breaking it up. In other words, it'll degrade the DNA. Thus, what was going to be transformed into a host cell by the virus, the bacterium's been able to protect itself now, so it's not infected. The catch was this: all living organisms, as I said many times over, have their genetic information stored in the form of DNA. In the case of prokaryotes, such as the Bacterium E. coli, okay, the DNA is double-stranded. It's a helix, but it's circular, okay, and it's DNA. Restriction enzymes are just looking for stretches of DNA it recognizes as restriction sites to bind to and cut. So what about the bacterial or the host cell's own DNA? Well, what the bacteria does is it attaches functional groups known as methyl groups. And that's what they're showing you here. They're showing you a methyl group. The bacterium attaches methyl groups to its own DNA, to its host DNA, as they're saying here, so that these enzymes are found to recognize these methyl groups as tags or identifications, letting the enzyme know, don't digest this DNA, don't destroy it, okay? Only bind to restriction sites located on DNA that doesn't have a methyl group. So the methyl groups serve as an ID to prevent the restriction enzymes from wanting to cut it up and digest it. So the bacteria, okay, does have a defense mechanism. But as you see in this figure, it's not one virus trying to infect one bacterium, okay? It's many viruses that have attached to the outer surface of the bacterium and that are all injecting their viral, say, DNA into the cell. So it's not a one-to-one relationship. So is it possible that these enzymes aren't able to keep up with the amount of DNA being inserted into them to where some of that viral DNA takes hold and does transform the bacterial cell into a host cell? Yes, it is possible. So what has been found is when a phage, bacteriophage, this is the abbreviation for a bacteriophage, everybody. On the test, I use bacteriophage, but just to let you know, if anybody ever talks about a phage, they're talking about a bacteriophage. That's the lay term it most often goes by. When a bacteriophage or a phage is successful in inserting its genetic information into a bacterial cell, what was discovered is that infected host cell will be taken into one of two life cycles, okay? It'll either be taken into a lytic life cycle or a lysogenic life cycle. I describe the lytic life cycle, which is down here, I describe it as an aggressive life cycle in a bad way for the host cell. The lysogenic cycle, I describe that as a type of life cycle that is passive, but it still doesn't benefit the host cell any. As a matter of fact, it's in a manner of speaking, a unique way of the virus indirectly infecting many cells over time by initially infecting only one. So as we go through this here, it's showing you when the bacteriophage attaches to the outer barrier of a bacterium, okay, from the head through the tail sheath will be injected its nucleic acid. Let's say it's a DNA phage. It inserts DNA, okay? Ignore this, what's happening to the host cell DNA. It'll insert its viral DNA, okay? If the bacteriophage causes the host cell to go into a lytic life cycle, okay, what that means is once the viral DNA has been inserted into the bacterium, transforming it into a host cell, that bacterium will immediately start performing DNA replication and gene expression to produce new viral particles. Okay, so you can see here, okay, copies of the viral DNA are being made. Genetic information stored in that viral DNA is being expressed to produce here. You can see the tail sheath, tail fibers here. You can see over here, all these different protein parts are being assembled, including the proteins that form the head or the caps around the DNA. The purpose for lytic has to do with the term lysis. To lyse means to rupture or break open. Well, what happens during the lytic cycle is once a certain number of viral particles have been synthesized and assembled together, what will happen is the host cell, it will be induced to lyse. In other words, it will rupture open, releasing the new viral particles. So theoretically, we go from one viral particle to many being produced. This is called the lytic cycle. This is why I describe it as an aggressive cycle. Once the virus has successfully infected the bacterial cell, transforming it into a host cell, the bacterial host cell starts the virus, and the host cell is ruptured or killed, or it undergoes lysis, releasing new viral particles. The other life cycle, called the lysogenic life cycle, even though I describe this as a passive life cycle, in a manner of speaking, you could say it's the scarier of the two. Okay. Sometimes what I found out is that when a phage infects bacterium, okay, once it injects its viral DNA, let's say it's a DNA virus, once it injects its viral DNA into a bacterial cell, transforming it into a host cell, that viral DNA somehow, some way is inserted. You can see here, they're showing you. It's inserted. Once that viral DNA is inserted into the bacterial DNA, notice here how it's inserted, now it's part of that circular bacterial DNA. We refer to that viral DNA as prophage DNA. Okay. Once that occurs, what was discovered to happen was that the genetic information, as well as the viral DNA, it doesn't undergo gene expression and it's not replicated. It sits there dormant, hiding within the bacterial DNA. It is a part of that bacterial circular DNA. Okay. Now, when this infected cell, when this host cell goes through binary fission, which is prokaryotic cell division, when it replicates its own circular DNA, well, the viral DNA or that prophage is part of that circular DNA. So you can see down here, the viral or that prophage DNA also gets replicated with the bacterial DNA. So when that one cell divides into two cells, now we go from one infected host cell to two infected host cells. And then when those two cells undergo binary fission, those two infected host cells, okay, result in four infected host cells. Everybody, this is comparable to HIV. HIV was discovered that when it infects a type of white blood cell called a T cell, it can cause that T cell to go into a lytic cycle, or it can cause that T cell to go into a lysogenic cycle, which I'll show you in a bit. Okay? If it causes those infected T cells to go into a lysogenic cycle, the person is HIV positive, but they do not have symptoms referred to as AIDS. Okay? It's comparable to what they discovered for bacteriophages. Okay? So again, the lysogenic cycle, I call it a passive phase, but I mean it's better. Okay? Now, could there be a factor that triggers bacterial cells in this life cycle, lysogenic, to subtly transform and switch over to the lytic cycle? Yeah, there is. I've never been able to come over anything specific other than in general some type of stress that would cause this infected host cell that's in the lysogenic cycle to suddenly switch over to this aggressive lytic cycle. It is known to happen, and it's the same thing for HIV. Okay. What I'm going to go into now has to do with animal viruses. Okay. There's a table in your textbook, okay, that lists 6 classes of animal viruses. Okay, there are going to be 2 we're going to focus on, and there are a total of 3 animal viruses. We'll focus on 2. One I've already gone over a lot of detail, influenza. I'm going to add a little more detail for HIV. Ebola, I'm just going to mention it there because you hear a lot about it. Okay, so all 3 of these viruses, okay, are animal viruses. When you look at their chemical structure, they're made up of nucleic acids. The nucleic acid is the same, single-stranded RNA. Now, the number of single-stranded RNA molecules can vary between the three. Okay, they all also chemically, structurally speaking, their chemical structures also consist of proteins. Proteins that form a capsid, proteins that form glycoproteins, and they all contain proteins that function as enzymes because they're RNA viruses. So they all have a prepackaged enzyme that'll help the host cell initiate the reproduction process of new viral particles. And then they all have a membranous envelope, except their overall shapes are not the same, such as Ebola virus. If you're familiar with it, you may have heard they refer to it as a filamentous virus along with Marburger virus. That's because of its shape. So when they classify these animal viruses, they classify them under one of 6 classes, simply class 1 through class 6 using Roman numerals. The Ebola virus and the influenza virus are both classified under class 5 viruses. They look nothing alike, but it has to do with the nucleic acids and what type of nucleic acid and how that acid is treated by the host cell when it initiates reproduction. Class 6 viruses, well, there's only one up to date, and it's HIV. Okay, so when we go over these, those are going to be the only 2 classes you're responsible for. However, this is the table that's in your textbook, and I'm still going to go over the first 4 classes because what I want to point out is, first of all, when they classify animal viruses, notice something. What they're focusing on initially is the type of nucleic acid. Is it an RNA virus or is it a DNA virus? Then the second thing they focus on is it double-stranded, DS, or is it single-stranded, SS? So notice that over here, this is what they're talking about. I already underlined these 2. Did I miss one? Yeah, this one. Okay. So when they classify animal viruses, first they're focusing on a nucleic acid. Is it a DNA virus or an RNA virus? If it's a DNA virus, is it a virus that contains or has its genetic information stored in double-stranded DNA? If it's a DNA virus, and it's a DNA virus that has its genetic information stored in double-stranded DNA, it's classified as a class 1 virus. Okay, so you can see some of these viruses are listed here, such as the herpes virus this here, smallpox virus. These are all viruses that have their genetic information stored in the form of double-stranded DNA. However, if it's a DNA virus, but its genetic information is stored in single-stranded DNA, then that animal virus is placed under class 2, such as the parvovirus. If you're like me and you have pets or you've had pets, this is where you become familiar with the parvovirus. A lot of times it can cost your pet their life. This virus, I find that at least for me with my pets, if they contracted this virus, it happened during the first 6 months of age. And I had it happen to a few pets, and we lost a couple of them due to this virus. Notice something, okay, for these 2 viruses, they're DNA viruses. One is double-stranded, the other has its genetic information stored in single-stranded. The last 4 classes of animal viruses, these are all RNA viruses. Look at class 3 viruses. These are viruses that have their genetic information stored in double-stranded RNA. In living organisms, you don't find double-stranded RNA. You may look at a figure and it makes you think the RNA is double-stranded, such as tRNA, but it's not. It's a single strand of RNA that folds up on itself. So it appears double-stranded, but it's not. Okay, but these are viruses that have their genetic information stored, okay, in double-stranded RNA, and you can see here they're giving you the one and only virus here. Now, if you look, class 4 viruses, okay, these viruses have their genetic information stored in single-stranded RNA. But notice something, class 5 and class 6 also have their genetic information stored in single-stranded RNA. So these last 3 classes, 4 through 6 of animal viruses, genetic information is stored in single-stranded RNA. But notice something, okay? For class 4 viruses, the genetic information that is stored in single-stranded RNA, once that virus inserts it into a host cell, the host cell recognizes that viral single-stranded RNA as mRNA and begins gene expression immediately. Your common cold viruses such as the rhinovirus, but notice something here. Here's your coronavirus. Okay. Right here, they're showing you Sars virus. SARS is an acronym or abbreviation for severe acute respiratory syndrome. Okay, the COVID-19 virus, okay, that we're having the pandemic with right now, its official name, from what I've come across, is SARS-CoV-2. So the COVID-19 virus is actually known as SARS-CoV-2 among the research community, and it would be classified under here, a class 4 virus. It's an RNA virus. It has its genetic information stored as single-stranded RNA, which actually serves as mRNA in the infected host cell. And here is its cousin, SARS, the one that was initially discovered. Now, these last two classes are the ones that you are responsible for knowing some information. Class 5 viruses and class 6 viruses. These viruses have their genetic information stored in single-stranded RNA, just like class 4. However, notice something. If an animal virus is classified as a class 5 virus, we go a step further. Yes, the genetic information is stored in ssRNA, but that single-stranded RNA serves as a template for mRNA synthesis. That's different from up here for class 4. Okay. Down here, the single-stranded RNA is not seen as mRNA by the host cell. Therefore, if you look here, there are the two viruses you're responsible for knowing that fall under here, Ebola and influenza virus. Therefore, for the influenza virus, I'm going to focus on this. Therefore, for the influenza virus, its genetic information, like Ebola, their class 5 virus, its genetic information is stored in single-stranded RNA. This single-stranded RNA is going to serve as a template for synthesizing mRNA synthesis. In other words, we're talking RNA replication, everybody. This RNA is a template for making more RNA. The cells of living organisms don't do that. They don't perform RNA replication. Therefore, that's why I was talking about the influenza virus. I'm going to use this figure here. Okay. This isn't to scale. Okay, but this is representing a class 5 RNA virus. Okay. Single-stranded RNA that serves as a template for the synthesis of mRNA. Consists of glycoproteins. Now, this isn't to scale, but let's say we're looking at the influenza virus. Instead of the Ebola, let's say it's influenza virus, okay? Making this up. The glycoproteins known as hemagglutinin would help attach the viral particle to the cell membrane of the host cell. The host cell would be a cell lining your respiratory tract through which air passes. Once that glycoprotein called hemagglutinin attaches to molecules on the surface of cells lining the respiratory tract, that will pull the membranous envelope close enough to the cell membrane of the host cell so that the membranous envelope, that phospholipid bilayer, and the cell membrane of the host cell, that phospholipid bilayer, fuse together, releasing the capsid into the ICF of the host cell. I forgot to I'll fix this here, but I meant to put this here. I had to add it in because it wasn't in the figure. Once the capsid is released into the host cell, the proteins that form the capsid will disassemble, releasing the single-stranded RNA that has attached to it the enzyme Rna Transcriptase. Okay. The cells of living organisms do not perform RNA replication. Somehow, some way, the influenza virus, which is not alive but not dead, knows this. Don't know why, but it does. So what happens? Since the cells of living organisms, or in this case host cells, don't perform RNA replication, no problem. The influenza virus provides an enzyme that will perform RNA replication. It will use that viral single-stranded RNA as a template to synthesize more RNA or to replicate that viral RNA. Except when it replicates that viral RNA, synthesizing a complementary strand, notice what happens. We end up with mRNA. So class 5 viruses, their genetic information is stored in single-stranded RNA. These viruses have to have enzymes contained in them, such as Rna Transcriptase, so that that enzyme can make a copy of that viral RNA template. But when it replicates that viral RNA template, that replicated RNA actually serves as mRNA. From this point right here, the host cell will take over the process of translation, producing the necessary proteins. That transcriptase will just keep performing viral replication, therefore making more of the virus's single-stranded genome, and eventually we have new viral particles exiting. Okay, so again, not alive but not dead, still trying to figure out how is it you know that host cells can't perform RNA replication. How is it you know exactly what enzyme to spot them with? The last class of enzymes are your class 6 enzymes, and there's only one. If I can find it here. Human immunodeficiency virus. That's the long name for a virus most popularly known simply as HIV. Okay? There's the virus HIV, and then there's a group of symptoms that can result from this viral infection that we call AIDS, acquired immunodeficiency syndrome. Listen carefully. HIV targets our white blood cells, specifically white blood cells called T cells. Now, some of the literature doesn't agree with this, but until I see it conclusively, the way I describe T cells is that they're the primary coordinators of your immune system and immune system attacks. In other words, they are the major players in ensuring your immune system can protect you. Therefore, if they are compromised, okay, your immune system is severely crippled, and now you are vulnerable to any type of pathogen, any infectious bacteria, any infectious fungus, other viruses. Okay, some of these pathogens are described as opportunistic pathogens, such as Candida albicans, a fungus associated with yeast infections. It's found on your skin. Somebody who's HIV positive and the virus is active, wiping out their white blood cells called T cells, it's been found that that fungus in those individuals can also cause pneumonia. Okay, so it cripples your immune system. Therefore, other pathogens take advantage of your body. There's no defense. And so you develop different disorders and diseases, which are all combined together and described as AIDS. So this is the only virus classified as a class 6 virus, HIV. It also has its genetic information stored in the form of single-stranded RNA, just like class 5 and class 4. Okay, but notice something. This is what really surprised researchers, and when researchers discovered this in the 80s, this is where they started to backtrack. Okay, they would go out there and start taking samples from various individuals, analyzing their white blood cells, because they discovered that this virus, okay, not only causes AIDS, but when it infects a person, it may not cause AIDS. It may sit there dormant. In other words, it may cause the white blood cells called T cells to go into a lysogenic cycle and hide out in a person. So the person's HIV positive, but they don't have AIDS. And it has to do with this here. It begins with this. HIV has its genetic information stored in single-stranded RNA, just like class 4 and class 5 viruses. Okay? And it serves as a template, just like class 5 viruses. But notice something. The single-stranded RNA from HIV, it serves as a template for DNA synthesis, not RNA synthesis. So when scientists discovered this, immediately, for those that were in tune, okay, they realized, oh my God, we've got a huge problem here. Why would a virus want to take its genetic information in the form of RNA and transform it or convert it back or into DNA? Well, it's so it could hide its information in the host cell's DNA. Okay. I'm not going to turn this information here, just to let you know this is slide 18 and 19. That's for your own viewing. I'm jumping ahead to here. Okay. So the only class 6 virus is HIV. Okay. Structurally speaking, HIV consists of a membranous envelope. They call it a viral envelope, but it's a membranous envelope. It does have glycoproteins. The most popular one at one time was called GP120, GP standing for glycoprotein. Okay, now HIV, make sure you know this, has 2 single-stranded molecules of RNA encased in a capsid, but also within the capsid is your enzyme reverse transcriptase. Okay. Notice it has as part of its name transcriptase, the ASE. I said whenever an enzyme has as part of its name transcriptase, automatically tell yourself it requires an RNA template. Except the first part of its name is reverse. In other words, this enzyme does the opposite of RNA polymerase. It goes in the reverse direction. RNA polymerase in chapter 17, it catalyzes the first step of gene expression, transcription. It uses a DNA template to synthesize mRNA. Reverse transcriptase performs a process called reverse transcription. Okay, so if you come over here, what reverse transcriptase does is it uses a viral RNA template to synthesize viral DNA. Once this enzyme reverse transcriptase has taken the viral single-stranded RNA and reverse transcribed it into single-stranded viral single-stranded DNA, it then hands over its viral single-stranded DNA. It then hands it over to the host cell so that the host cell's own DNA polymerases can replicate the complementary strand. So with the help of reverse transcriptase prepackaged into the HIV, this enzyme helps catalyze the reproduction process by first taking its own viral RNA and reverse transcribing it into single-stranded DNA, handing it over to the host cell so it can replicate a complementary strand. Now we have double-stranded viral DNA that is then inserted into the host cell's DNA, where now we call it a provirus. So this is an HIV provirus. Here it is here. Okay. Now, one of two things happens to the infected cell. Either this provirus DNA, okay, sits there dormant, but every time the host cell divides, it not only replicates its own DNA during S phase, it replicates HIV's provirus DNA. Now, either right away or due to some stress, eventually what can happen is that host cell, okay, that infected T cell, can be caused to go into a lytic cycle where the genetic information that is inserted into the host cell DNA, it's now expressed, resulting eventually in reproduction of new HIV viral particles. Over here they're showing you, this is the membrane of a white blood cell. Here's HIV, it's infecting the white blood cell, transforming it into a host cell. And notice something, I said lytic cycle, but technically speaking, okay, I don't know if they actually describe it as a lytic cycle. I've never seen a variation on it, but it's showing you the virus can exit the white blood cell without actually causing the host cell to lyse. Okay, my understanding is eventually the host cell will lyse and be ruptured open, but it's showing you it can just come out drop by drop, reproducing. So this really scared the science community and the medical community. It's like, oh my God, this virus willingly wants to take its RNA and convert it to DNA and hide it in the host cell's DNA. The catch is this, researchers, okay, they try to see the silver lining in everything. So if you recall in lab, we didn't get to do it in person, but for the GMO lab, you performed a process called PCR. Well, eventually what happened is scientists isolated the genetic information stored in that single-stranded RNA, okay? And in vitro, they were able to take that genetic information that coded for reverse transcriptase, and in vitro, in a tube, they were able to produce this protein enzyme called reverse transcriptase and market it, sell it to labs, so that labs could perform a variation of PCR called reverse transcription Pcr. So now labs could extract mRNA from a cell when they wanted to study gene expression and alterations in gene expression, and they could use this enzyme reverse transcriptase to take that mRNA, copy it into DNA, and then use your gel electrophoresis and so on. So if you ever hear a lab technique called RT-PCR, they actually use this enzyme, which came from HIV. Okay, so scientists are always trying to figure out, okay, this is a very evil virus, but let's see if we can use it to our advantage, at least part of it. So here it's showing you T cell. There are different types of T cells. Specifically, it's what we call helper T cells. You can see there are many, many viral particles of HIV attached. Some of the glycoproteins recognize molecules on the surface of the T cell, and that's what allows them to anchor themselves to it. You can see over here, it's showing you many of these HIV viral particles infecting a single white blood cell, single white T cell. Okay, so this showing you the virus is trying to infect the cell. I forgot to point this out real quick, even though I'm not going to test you on it. This figure right here, the reason why I put it here is because it's actually not showing Ebola trying to infect the cell. Okay, it's actually showing the Ebola virus, if you look here, growing out of the infected cell. This is in the lab. So it's showing you all these little blue strands represent the virus. So listen carefully for vaccines. When they had the Ebola outbreak a few years ago, I don't watch CNN and Fox and all that and MSNBC much, but I happened to just scan across CNN, and they had a news reporter there interviewing a doctor, and he told the doctor, well, why don't you just use antibiotics to fight this Ebola virus infection? And the doctor paused for a second, and he was real nice to the doctor, and he politely let the guy know that antibiotics are used to treat bacterial infections. They don't do anything to a virus. Okay? The best way to combat viruses, and it's not perfect, and I'm not advocating for it, I'm just telling you the best way so far to date to combat viral infections is through preventative measures using vaccines. Okay? Some people think when they receive a vaccination that they're receiving some type of chemical drug like comparable to ibuprofen or something, or an antihistamine, like with Claritin or something. No. When you're given a vaccine, you are either giving an attenuated version of a virus, which means you're getting the viral particle, but it's been treated to where structurally it's intact, but it's not active, or you're being given pieces of the virus, such as with the flu vaccine. With the flu vaccine, it contains pieces of the influenza virus, specifically hemagglutinin and neuraminidase. So vaccines are harmless derivatives of pathogenic microbes that stimulate the immune system to mount defenses against harmful pathogens. So what is happening when you're being given a vaccine? Okay, you're being given an attenuated form of the vaccine, either the virus itself, which is structurally intact but harmless, or pieces of the virus, which have also been treated so that they're structurally intact but harmless. They're injected into you so your immune system can take a fingerprint. Our immune system relies heavily, okay, on recognizing what belongs in you versus what's foreign based on molecular structure. And what our immune system will do is when it recognizes something as foreign, it not tries to destroy it or destroys it. It takes a fingerprint, okay? And when it takes that fingerprint, it also comes up with a recipe on how to remove that molecule. And then it usually will store the recipe on how to destroy that foreign object, okay? It'll save it in its memory bank. Okay, now it varies from virus to virus, but this is the purpose of the vaccines. They're preventative measures. Now they are working on antiviral drugs. So when HIV came out in the 80s, the antiviral drug they came out with was AZT. AZT, the key component with AZT is it consisted of DNA nucleotides, but one type of DNA nucleotide it had in it was a mutated form that would interfere with the synthesis of DNA. Thus, the thought process was, or the theory was, when the virus tries to reverse transcribe its single-stranded RNA into single-stranded DNA, by administering AZT, these mutated nucleotides would inhibit the replication, or the production or synthesis of the viral DNA. That's the thought process. I haven't checked for the more recent drugs, although I do know there's 2 out on the market now. Okay, this is the last part. Are there any questions up to now? This is the last part we're at. Okay, prions, listen carefully. Right when researchers thought, okay, the smallest infectious agents are viruses, they started noticing that there were sheep that were a little neurotic to the point that they were constantly gnawing and rubbing up against things, scraping off their wool. Or there were cows that started acting weird, might drop their head down, kind of swam left to right, or human beings that seemed like they were displaying some type of neurological disorder, but it wasn't Alzheimer's, it wasn't MS, it wasn't Parkinson's, it wasn't schizophrenia. So eventually what they figured out is there's actually an infectious agent that's smaller than a virus, much, much smaller than a virus. They call these prions or prion proteins. The abbreviation for prions or prion proteins is PRP. Okay. Now down here, if you notice, I put PRP superscript C versus PRP SC. PRP with the superscript C that is what is described as normal prion protein. What was found is that animals, okay, not just human beings, but animals, that the nervous system in particular produces proteins called prions or prion proteins. They're found located in the membrane of cells of the nervous system. Not really sure what the overall function is, okay, but they are required. So when these proteins are synthesized and when they're being put together and everything and inserted into the cell membrane, if they're normal functioning proteins, sometimes I'll abbreviate them PRPC, cellular prion protein. However, what they found is that sometimes, okay, sometimes the protein will misfold. So in the textbook, they kind of simplify it. But sometimes what happens is the protein will misfold. So in literature that's more technical, they'll still use the PRP for prion protein. But if it's a disease-causing prion protein, they'll use a superscript such as SC. Okay. What they found is, for reasons unknown, the normal prion protein will sometimes be misfolded during post-translational modifications. Okay, before it ever makes it into the membrane, it'll be misfolded, becoming a disease-causing prion protein. Okay, if it's causing scrapies in sheep, they'll use PRPSC. Okay, what they found with this misfolded protein is, even though it's not understood what's causing the misfolding of this protein, which is normally produced by cells of the body, especially in the nervous system, but that this misfolded protein, when it comes in contact with normally folded proteins, it will induce these normally folded proteins to also become misfolded. The second characteristic of this protein is that it is, in a manner of speaking, sticky. So when it causes other normally folded proteins to become misfolded, they start to aggregate or clump together. Okay. This is something you see comparable to, say, Alzheimer's. When they study people with Alzheimer's, they find that, say, beta amyloid, you'll hear talk about beta amyloid plaques, which are basically dead zones in a person's nervous system, especially in the brain, where you have a high concentration of proteins called beta amyloid that are sticking together to each other and in neurological problems. Well, this is what starts to happen. They start to stick to each other. These disease-causing prion proteins start to stick to each other or agglutinate within the nervous cell, causing the cell to dysfunction. Thus, that impairs nervous system function, and you get something like a sheep that for some reason starts gnawing at its skin or rubbing up against things, scraping off its wool. Okay, we call it scrapies in sheep. But when the disease-causing prion protein forms in cows or bovines, okay, technically they call it bovine spongiform encephalopathy, or BSE. In lay terms, it's known as mad cow disease. Okay, this is the brain of a healthy cow, and this is one that has the disease-causing prion proteins in it. You can see it's causing the breakdown of nervous tissue, therefore it's causing neurological functions. These misfolded protein, disease-causing prion proteins, okay, they also can cause neurological problems in human beings. Okay, when that occurs, whenever these disease-causing proteins, okay, infect a person's body, okay, the neurological symptoms it causes is referred to as Creutzfeldt-Jacobs disease, or CJD. Now, the catch with this, the reason why the disease-causing prion proteins are contagious, okay, is that these proteins are highly resistant, these misfolded disease-causing proteins are highly resistant to denaturation. Therefore, you may have heard, and this happened in England roughly in the early 2000s, there was an outbreak on the media of mad cow disease, or bovine spongiform encephalopathy, which was caused by disease-causing proteins. The farmers in England said, hey, look, okay, when the government told them you're going to have to wipe out your entire cattle, every cow you have, they said, no, not all of them are infected, not all of them are exhibiting symptoms. But the health department said, no, you don't understand. Okay, if anybody consumes any diseased tissue from one of these cows, even if it's not showing symptoms, they may be in the early stages, it's not detectable. But if anybody consumes any tissue that contains this disease-causing prion protein, what will happen is the prion protein will pass right through your digestive tract, never being denatured, especially by the acidic environment inside your stomach, will not be broken down by enzymes in your small intestines, and therefore you could absorb it into your own body, where in turn it could cause a disease state called Creutzfeldt-Jacobs disease. So whenever it's found that animals are infected with a disease-causing prion protein developing scabies or mad cow disease, they have to wipe out the entire herd to prevent it from being transmitted to human beings. So as of right now, the smallest infection agent is a misfolded protein called the prion protein, or PRPSC. Okay. Yes, we finally got to the end of the chapter. End of the, as I said, I don't cover 20 and 21 right now because most of that's covered in the lab. I'm going to go ahead and stop the recording here.